Have you ever wondered what it would be like to be immune to diseases like cancer or HIV? What if we could wipe out genetic disorders like Huntington's disease or cystic fibrosis before a child is even born? Sounds like science fiction, right? Well, with a revolutionary technology called CRISPR, it's closer to reality than you might think. But this incredible power brings with it some of the most profound ethical questions humanity has ever faced. Where do we draw the line? Today, we're diving deep into the complex world of gene editing ethics. So, what exactly is gene editing? Imagine the DNA in our cells is like a massive encyclopedia, containing all the instructions that make us who we are. Gene editing tools, especially CRISPR-Cas9, act like a pair of molecular scissors combined with a find and replace function. Scientists can use it to find a specific gene, a single word in that encyclopedia, cut it out, and even replace it with a new one. This has been a game changer in biological research. Let's start with the undeniably positive side, treating diseases. This is often called somatic gene therapy. Somatic cells are all the cells in your body that aren't sperm or eggs. Editing these cells would only affect you, the individual patient. For example, scientists are already running clinical trials to treat sickle cell anemia. They take a patient's own blood stem cells, use CRISPR to fix the faulty gene that causes the sickle shape, and then put the corrected cells back into the patient. The results so far have been incredibly promising, offering a potential cure for a lifelong, painful disease. The same approach is being explored for blindness, muscular dystrophy, and even certain types of cancer. Ethically, this is the least controversial application. It's essentially a new, highly advanced form of medicine, aiming to cure suffering in existing people. Most of us would agree that if we have the power to fix a debilitating disease, we have a moral obligation to do so. But here's where things get much more complicated. What about germline editing? This means editing the DNA of embryos, sperm, or eggs. The key difference here is that any changes made are not just for one person. They repass down through generations. These changes become a permanent part of the human gene pool. On one hand, this could be the ultimate form of preventative medicine. We could eradicate hereditary diseases like Tay-Sachs or Huntington's disease forever. A family that has been haunted by a specific genetic illness for generations could finally have healthy children, and their children's children would be free from that curse as well. This is a powerful, compassionate argument. Why treat a disease in every generation when you could prevent it from ever occurring again? However, the idea of editing the human germline opens a Pandora's box of ethical dilemmas. The first major concern is safety. We're talking about altering the very blueprint of a human being. What if we make a mistake? CRISPR is precise, but it's not perfect. It can sometimes make off-target edits, changing parts of the DNA it wasn't supposed to. The long-term consequences of these unintended changes are completely unknown. We could inadvertently create new health problems that might not show up for years, or even generations. We are literally experimenting on future generations without their consent, which is a massive ethical red line for many people. The technology is still new, and many scientists argue that we are nowhere near ready to use it on human embryos for clinical purposes. Then we have the slippery slope argument. We start by using germline editing to prevent terrible diseases. That seems noble. But where do we stop? What is the difference between fixing a gene that causes a disease and enhancing a gene for a desirable trait? If we can prevent deafness, should we also be able to enhance hearing? If we can correct poor eyesight, why not give our children perfect vision? It's a blurry line between therapy and enhancement. This leads to the scary concept of designer babies. Imagine a future where wealth determines your genetic makeup. The rich could afford to give their children genetic advantages, higher intelligence, greater athleticism, a longer lifespan. This would create a genetic divide, a biological class system. Society would be split into the genetically enhanced and the naturals. This could exacerbate existing inequalities to an unimaginable degree, creating a world that feels more like a dystopian novel than a utopian dream. The very idea of shared humanity could be threatened if we are no longer playing on a level genetic playing field. Is it fair to engineer advantages for some, while others are left behind? Furthermore, who gets to decide what is a defect and what is just a natural human variation? Many people in the deaf community, for example, don't view deafness as a disability to be cured, but as a cultural identity with its own language and traditions. From this perspective, editing out the genes for deafness could be seen as a form of eugenics, devaluing certain ways of being human. It implies that there's a correct way for a human to be, and anything that deviates from that norm needs to be fixed. This could lead to a loss of human diversity, which is one of our species' greatest strengths. We risk creating a monoculture of what we deem to be perfect and losing the rich tapestry of human difference. This issue came to a head in 2018 when a Chinese scientist, He Jiankui, announced he had created the world's first gene-edited babies. He used CRISPR on twin embryos to try and make them resistant to HIV. The global scientific community reacted with outrage. He was widely condemned for conducting a dangerous and ethically irresponsible experiment on human beings, bypassing all established ethical guidelines and protocols. The long-term health of those twin girls is still unknown. This event was a major wake-up call, showing that this isn't just a theoretical debate anymore. The technology is here, and rogue actors might use it without regard for the consequences. So where do we go from here? The debate is ongoing, and it's one of the most important conversations of our time. Most countries and scientific organizations have called for a moratorium on clinical germline editing, meaning a temporary ban until we can have a broad societal consensus on if and how it should be used. There are international committees working to establish clear guidelines and regulations. The key is to involve everyone in this conversation, not just scientists and politicians, but ethicists, religious leaders, disability advocates, and the general public. This is a decision that affects all of humanity, and everyone deserves a voice. The potential of gene editing is breathtaking. It offers hope for curing some of our most devastating diseases. But with great power comes great responsibility. We stand at a crossroads. One path leads to a future free from genetic disease, but it's a path fraught with dangers like unforeseen health consequences, social inequality, and a loss of human diversity. The other path is one of caution, where we prioritize safety and ethical considerations, perhaps moving more slowly, but more responsibly. What do you think? Should we use gene editing to eliminate diseases, even if it means altering the human germline? Where would you draw the line between therapy and enhancement? This isn't just a scientific question. It is a question about what kind of future we want to create for ourselves and for all the generations to come. Let me know your thoughts in the comments below. This is a complex topic, and hearing different perspectives is crucial. Thanks for watching, and if you found this video thought-provoking, please give it a like and subscribe for more deep dives into science and ethics.